The next chapter we're going to look at is chapter 8, which deals with cell-mediated effector mechanisms. Now, we know that predominantly we're talking here about T cells and things that are intracellular. What things do you need to take home as far as cell-mediated immunity for the boards? We need to know what we're talking about with cell-mediated immunity, which is our response to intracellular pathogens. What are the effector cells and how do they kill? And how do we regulate this response? Now, cell-mediated immunity, it kind of completes the package here, right? We have our antibodies that were our response to extracellular pathogens, and now we have our T cell response, which is our response to intracellular pathogens. We're going to look predominantly at 3 major cell types here, okay? And they're all aided by the T helper 1 cell. Okay, if we go back to remembering our dichotomy, we had the TH0. Depending on the response, we differentiated into TH1 and TH2s. We just talked about TH2s and the role in promotion of humoral immunity. And now the TH1 cells, of course, we're going to be looking at as far as promoting cell-mediated immunity. All right, which cell types are important in cell-mediated immunity? You don't maybe think of the macrophage very often, but it's a really important cell type in cell-mediated immunity, and we'll talk about that. Usually we think about cytotoxic T cells as far as cell-mediated immunity because they blow up target cells, as do the natural killer cells. So we're going to look at these different mechanisms of cell-mediated immunity. Now, macrophages are interesting, okay? And macrophage killing, when are we going to use macrophage killing? We're going to use it for things that are kind of sneaky, okay? In microbiology, we talked about the facultative intracellular pathogen, the best example of which is tuberculosis, okay? And with facultative intracellular pathogens, what do we know that they do? All right, what does TB do? Macrophage takes up tuberculosis. Does TB care? Not really, because TB escapes killing from the macrophage. So now it's hiding out in an immune cell, escaping our normal mechanisms of immune defense. How do we kill it? Okay, it's really still going to go with the macrophage. The macrophage has some tricks up its sleeve for helping to eliminate these facultative intracellulars. Now, it's cell-mediated. It happens through the TH1 response, and the most important cytokine you need to know for this is interferon gamma. Okay? Interferon gamma, remember we talked about this earlier, takes the macrophage from kind of a normal, just everyday lymphocyte into the Incredible Hulk. So it becomes this very efficient killing machine. Now, your tumor necrosis factors are also important, but interferon gamma is very important. Okay? Now, the killing occurs intracellularly. How does that happen? Okay, the tuberculosis is hanging out inside of a macrophage. Well, what happens is the macrophage knows, okay, that it has something inside of it. And there's a couple of ways the macrophage is going to deal with this, all right? The first that we know and we always talk about with TB, if a macrophage can't directly kill something, what does it do? It seals it off in a granuloma. So we have granuloma formation whenever we have facultative intracellular pathogens, okay? The other thing that happens here with the macrophage is it's extremely agitated now. It's in this hyper-aware state. It's got its interferon gamma. It's a killing machine, but it can't do its job. It's not being able to kill things that it takes in through phagocytosis. But it has upregulated all of these toxic oxygen radicals, nitric oxide, cytokines. It's ready to go. So what happens? It just starts releasing those chemicals. And you're going to start to get necrosis of the surrounding tissue. It's just going to spew it all out. So you're going to get necrosis of the surrounding tissue. Now, is this a good thing? Well, it's going to kill some healthy cells, but it's also going to kill cells that are harboring those fugitive bacteria. Remember, they're hiding out. They're not being killed by the macrophage. So if the macrophage can take what it would normally use within a phagolysosome to kill and now just release it, it's so toxic, the cells that come in contact with it are going to die. And you will kill some cells that are healthy, but the main job is that you're going to kill those cells that have those pathogenic bacteria within them. Okay? Now, macrophage killing, the macrophage is really kind of the hitman in this DTH response. Okay? It's a measure of your T helper 1 function. We'll talk specifically about the DTH response, which is delayed type hypersensitivity, a little bit more when we get to the hypersensitivity chapter. But what's really important about DTH is that the DTH response is your normal response against facultative intracellular pathogens. Okay? Now, cytotoxic T cell killing, this one's pretty straightforward. In our grand scheme, remember, of immunity, we've got this whole little mob family set up. We've got the T helper cell as the mob boss. They direct everything. They don't get their hands dirty. They communicate through cytokines. The cytotoxic T cell is the killer. It's the hitman. It's going to take advice from the helper cell, and it's going to use that to go kill a cell that has a virus in it, or maybe a cell that has transformed. These are our CD8 positive cells, okay, recognizing class 1 MHC, okay, plus peptide. It could be a viral peptide, it could be an altered self peptide, okay. Now, the T helper 1 cell is providing the cytotoxic cell. The main signal is IL-2. That takes it from your normal kind of CTL to now an actual killer cell that is capable of functioning as a killer cell. Okay? Interferons, one of the things that interferons do is it helps to increase the expression of MHC, and you'll see this in both class 1 and class 2 MHC as well. Okay? So we're also going to see an upregulation of the MHC. Now, how do these cells actually kill? These are the steps involved in killing. You have to attach to the target cell. It does that through its T cell receptor, binding to class 1 MHC and antigen. What helps it recognize class 1 MHC is, of course, CD8. And then you have your LFA1, okay? That is kind of your tethering molecule. Activation, what does that mean? Well, your cytotoxic cells have all these granules in them. And now those granules are going to start to move, and they're going to start to line up between the cytotoxic cell and the target cell. Then we're going to release the contents of the granules. That's going to kill the target cell. And then we're going to detach so we can go do it to another cell. Okay? Now, what are the components within the granules? So this just kind of lists some of the granule components. Now, like any good hitman, it's not just going to do one thing. Because what if it misses? If it fires a gun and misses and walks away, the target might still be alive. All right? So what CTLs do is the first thing they're going to release, well, they're actually all released together, obviously, but the first function is going to be with the perforins. Perforins, just like the name suggests, perforate the membrane of the target cell. So it punches a hole in it. Would that kill the target cell? Probably. It would probably die. Not good enough for the CTL. Once it punches a hole in it, it pumps it full of toxic chemicals. What are the toxic chemicals? You've got degradative proteases that are going to chew up everything within the cell. And if that's not enough, you have cytokines that are going to go ahead and tell the cell, you know what, you should just go ahead and kill yourself by apoptosis because you're going to die anyways. So we punch holes in it, we pump it full of toxic chemicals, and then we leave, okay? On a good day, we've all had good days and bad days. On a really good day, when everything's going your way, we don't have to go through all of these different mechanisms. On a good day, a cytotoxic T cell can recognize a molecule on the target. So instead of going through all of that degranulation, it recognizes a fast molecule by way of its fast ligand. And once those two molecules interact, it just tells the cell to go kill itself. All right? That's much cleaner, much less messy for the cytotoxic cell. It doesn't have to, reconstitute its granules and all that stuff, so it's much simpler. So this is really just a diagram of cytotoxic T cell killing. In the first part of the diagram, you can see the cytotoxic T cell binding to the target cell. And what happens, of course, is the granules will get activated. You'll get degranulation, which you're kind of seeing here. And then this is kind of a blown up version of that. So you can actually see that we've punched holes in the membrane. We're releasing the granzymes. We're releasing the cytokines that are going to cause that cell to die. Okay? In the alternative situation with the fast ligand situation, what we're going to get eventually is the release of TNF. And when TNF binds to its cognate receptor on the target cell, that's going to be really the principal mechanism that causes it to die by apoptosis. So those are your two different mechanisms of CTL killing. And now I'm going to show you an animation of CTL killing by way of the first mechanism with perforins, etc. Okay, so let's look here. We've got a viral infected cell. So we're going to say we have a virus that's going to infect our target cell here. Okay, so it gets into the target cell, starts to replicate, becomes an endogenous antigen that gets presented on a class 1 MHC. Okay, now we're going to get presentation of that antigen to a cytotoxic T cell. So here's our T cell receptor. And what would this molecule represent? It represents CD8. Attachment is the first step. What's the second step? Activation. Activation, the granules are going to realign at the interface between the cytotoxic cell and the target cell. Third step, we're going to degranulate. So we're going to blow up the target cell. Okay? We're releasing the contents of the granule. We're blowing up and killing the target cell. What's the third or the final step? The fourth step is detachment. Now this cytotoxic T cell is going to fill up its granules again. It's going to realign the granules, and it's going to be able to go do that to another target cell. Okay? So that's your predominant mechanism of cytotoxic T cell killing. Now, flip side, let's look at natural killer cells. Natural killer cell killing, the important thing here, and just to make it very simple for you, the killing mechanisms, i.e. perforin, granzyme, cytokines, are exactly the same as a cytotoxic T cell. Okay? Now, what's the difference? Well, remember, natural killer cells kind of sort of look like a lymphocyte, but not really. Why are they not really truly lymphocytes? Because they don't specifically bind antigen. Okay? So, how are they going to recognize a target cell? They've got two different ways that it does that. Once it binds the target cell, all of the mechanisms it's going to use to kill the target cell, same as what we talked about for the cytotoxic T cell. Okay? Now, granzymes, perforin, just what we talked about for the cytotoxic cell. Some of the cytokines are similar. IL-12 and the interferons, alpha and beta, also help the natural killer cell. Okay, they're the cytokines the natural killer cell is looking for. What, though, is the natural killer cell's target? How is it going to know what to kill? Well, all nucleated cells should have class 1 MHC on it. But during certain circumstances, cells don't make class 1 MHC. Transformed cells can drop expression of class 1 MHC. Viral infected cells, we can either mask it or downregulate it so cells can't express class 1 MHC. Why would a virus do that? Virus would do that to get around that cytotoxic response. But now we've got the natural killer cell response. Natural killer cells kill cells that do not have class 1 MHC. So that's one of the ways that it finds its target. Okay, now what are some of the specific markers on natural killer cells that you should probably know? Just remember the 6s. You have CD16 and CD56. So later when we start walking through the histograms, we throw up something with those markers. You should know that those identify a natural killer cell. Okay, so what's the situation here with natural killer cells? Natural killer cells kind of go around and just kind of survey things. They're like, you have class 1 MHC, you have class 1 MHC, you're fine. And it doesn't touch cells that have class 1 MHC because, number one, it's supposed to. And number two, if it does have a viral infection, it knows that the cytotoxic cell will take care of it. But it's just kind of like duck, duck, goose. It's like class 1, class 1, class 1. Oops, no class 1. And if a cell doesn't have class 1 MHC, the natural killer cell will kill it. Okay? So it has what's called an inhibitory receptor. And that's what binds class 1. That's what it says. Okay, you have class 1. I won't kill you. All right? Down at the bottom here, okay, we have this activation signal with nothing binding to the inhibitory receptor. So no class 1. That's the signal the natural killer cell needs to go ahead and kill the viral infected cell. So that's one of the ways that a natural killer cell can kill a target cell, okay, without specifically recognizing antigen because it doesn't have that capacity. Now, the other mechanism, remember we said we see antibodies again. The other mechanism is through ADCC, or antibody-dependent cell-mediated cytotoxicity. All right, and while we always really talk about this with the natural killer cells, there are some other cell types that can mediate ADCC, but predominantly we're talking about the NK cells, which is the only antibody isotype that can mediate ADCC. Is, of course, IgG. Okay? Same components are going to be there to kill the target cell once that natural killer cell degranulates. Okay? So in this particular diagram is simply showing the antibody binding to all the different cell types. And we're going to focus specifically on the natural killer cell. You have on the natural killer cell, you have an Fc receptor that specifically recognizes the Fc portion of IgG, okay? And that IgG is bound to a target cell. And I'll show you why that might be. I'll give you an example. Once that happens, the natural killer cell degranulates, punches holes in the target, and then is able to kill the target cell. All right? Antibody gives natural killer cells a little bit of specificity because the antibody is specific for antigen. Okay? Now, here's another animation of ADCC so I can show you why on earth an antibody would be binding to a cell. Okay? Same situation. We have a natural killer cell, though here we have a target cell. The pink cell is your target cell. Here comes our virus again. Now, an enveloped virus, okay, if we think about enveloped viruses, an enveloped virus has to, during its replication process, put viral-specific proteins in the membrane of the cells that they infect. So let's keep that in mind as we go through here. So this particular virus gets into a target cell, begins to replicate, and will cause this target cell to express viral-specific proteins. Now, are those proteins self? No, they're non-self. So antibodies can certainly recognize those viral-specific proteins, bringing them to an Fc receptor on, in this case, a natural killer cell. And now the natural killer cell is going to do exactly what the cytotoxic cell did. is going to punch holes in the membrane, pump it full of toxic chemicals, and cause it to die. Okay, so those are your two different mechanisms of natural killer cell killing. One, if we don't have class 1 MHC, and two, with the addition of antibodies. Now, in one instance, and you don't want to get confused, because if they ask you about ADCC, predominantly they're going to be asking you about IgG. But in the case of a parasite, okay, IgE can mediate this reaction, the ADCC. So unless they're very, very specific, you're always going to go with IgG. Okay, but that's just kind of one of the exceptions to the rule. Now, your overview of cell-mediated immunity, and we'll bring back kind of the Th1, Th2. Of course, with humoral immunity, we were talking about going down this Th2 pathway, which led to the production of antibodies and class switching. Okay, but now with cell-mediated immunity, we're really talking about this T helper 1 response. Okay, your cell-mediated immune response. Probably the two most important cytokines that are made by the Th1 cell for promoting CMI is interferon gamma and then interleukin 2. Okay? Now, what does interferon gamma do? Remember, it helps with macrophage activation. It also helps with the activation of some of these other cells, but so does IL-2. IL-2 is really the cytokine that you're looking for if you're a cytotoxic T cell. They're going to help kill a viral infected cell. And then, of course, your natural killer cells are looking for a cell with no MHC, or with the help from this side of the diagram, a little help from an antibody can help it specifically recognize a target cell. Okay? Once macrophages become activated, they also can produce a host of cytokines that have a big role in the inflammatory response. Okay? Now, that's kind of your overview, and just specifically in these next couple of tables, it just really kind of summarizes for you some of the different mechanisms in cell-mediated killing, differentiating all the different cell types that we talked about. It lists the different CD markers. That's very important for USMLE because, again, sometimes instead of saying cytotoxic T cell, in fact, most of the time, they're just going to say a CD8 positive cell. So you definitely need to know the CD markers, okay? The antigen recognition is the only specific for the cytotoxic T cell. In a natural killer cell, specificity is provided by the antibody or the lack of class 1 MHC. In a macrophage, the killing is nonspecific. Remember, we know that there's an infection somewhere, we know it's hiding out, and we're going to nonspecifically kill cells in the area because we're activated. Okay? Now, is MHC required? Of course, it's always required for cytotoxic T cells. Lack of MHC is required for killing from a natural killer cell. Okay? And MHC is not involved with macrophages. Now, the effector functions and molecules are the same for cytotoxic T cells and natural killer cells. A little bit different in the macrophage. Remember, we've got all of our pro-inflammatory cytokines, including IL-1, IL-6, and TNF-alpha. We've got our enzymes. Remember NADPH oxidase, myeloperoxidase, nitric oxide, and then eventually the production of these oxygen radicals. Okay, so those are your effector molecules. We discussed this, but here's a clinical correlate as to why natural killer cells are very helpful in killing cells that are infected with viruses. There are viruses, okay, that can downregulate class 1 MHC, and in that case it's very helpful to have natural killer cells. There are other viruses that downregulate MHC but also produce decoy molecules that can fool the natural killer cell by providing kind of a decoy that looks like class 1, but in that case we still have antibody-dependent cellular cytotoxicity. Now, RNA viruses are very, very rapidly replicating viruses, especially if you think about poliovirus, for example. They replicate very, very rapidly until finally the CTLs are able to kill those cells, okay? But in this particular case, if something replicates rapidly and, in the case of poliovirus, lyses a cell, antibodies are also going to be important in that particular response as well, okay? So that wraps up Chapter 8, Generation of Cell-Mediated Mechanisms.